Well, I mean, that's a $60 million question that especially researchers, a lot of people ask us. You know, LipoGel is a proprietary formulation. Let me ask it or let me propose this. The important or salient point to patients and clinicians is that it does inhibit biofilm. And that has been proven through the testing that the U.S. government did, as well as the research that we've done and published in the literature. Now, LipoGel was not, its composition was not developed haphazardly, okay? Things were chosen for reasons. And some of the key areas that we were looking to target were to block the initial attachment of bacteria, which is the initial step in biofilm formation. We wanted to, through quorum sensing type inhibitors, we wanted to inhibit the metamorphic changes from the planktonic bacteria to the biofilm phenotype state. There's actually some chemical signals in there to stimulate the reversion from the biofilm phenotype back to the planktonic phenotype. And finally, also there are lipids in there that we know inhibit toxin production by specific bacteria species. So that's a general strategy that's been done. And while the evidence for that is in our own internal research, there is outcomes-based evidence that is already published in the Journal of Wound Care, JAMA, et cetera, based on the studies that we've done with these to support actually the outcomes-based rather than the specific mechanism. And again, you know, how does aspirin help prevent atherosclerotic plaques? Okay, well, basically we have a mechanism theory for that, and there's several out there, one of which actually is biofilm reduction. As a matter of fact, salicylic acid is a fairly potent anti-biofilm agent. But, you know, scar tissue, anti-inflammation, all of these mechanisms proposed for aspirin is acetylsalicylic acid, excuse me, is really just proposals. And all the things that I cited for you are proposals that we try to go after strategically with things that you know, would be accepted by the FDA as you know, not a drug. So I've proposed some mechanisms for you, but at the end of the day, the outcomes and the fact that it does work is probably the most salient and important point. I just felt that you know, I list those for your benefit. That's a working hypothesis, and it certainly was designed for that. But exactly what's going on a molecular level with bacteria is, you know, we just hope to understand that better. We're never going to fully understand that.
So how effective is the LipoGel?
Well, from that standpoint, I don't think it would be appropriate for me to give any subjective answer to that. But all they need to do for that is to actually look to the literature, the study that we did with LipoGel by itself, as well as LipoGel RX guided by DNA. Those studies are done, at least the last one was published in May. That's done on about 1,400 patients, so it's a pretty large study. And you know, they can see the results, the outcomes from the self, and you know, those results are pretty dramatic. Depending on the wound type, we're talking about anywhere from 2 to 5 times faster healing rates. And as far as absolute healing, we were looking at about twice the healing. And no matter what period of the study you looked at the different treatment arms, how many patients were healed at that point. It was around twice as many. At least now we're talking about DNA-guided LipoGel RX. So I think that when we talk about efficacy, we really need to stick to the protocol-driven studies. The objective studies has been done rather than, you know, my personal opinion.